Hello, and welcome to Oncology for the Inquisitive Mind. Each year, the prestigious Beverley Alt Scholarship provides senior fellows at the Kinghorn Cancer Centre in Sydney an opportunity to enrich their educational and career training activities. This fellowship honours the life of Beverley Alt and the compassionate care she received at the Kinghorn Cancer Centre. As such, our very own Dr. Josh Hurwitz abandoned me to go gallivanting in the United States of America. He was able to attend the San Antonio Breast Cancer Symposium in Texas, as well as engage with some of the brightest minds in cancer care and research at the Dana Farber Cancer Institute in Boston. Meanwhile, I was left to freeze in one of the coldest Australian summers on records. No, I'm not bitter. Oncology for the Inquisitive Mind, supported by the Kinghorn Cancer Centre and the Beverley Alt Scholarship, is incredibly honoured to present a series of interviews with specialists who have influenced the course of medical oncology on both a global and personal scale, providing the promise of innovative, personalised medicine. In this episode, we stay at Boston's famous Dana Farber Cancer Institute as Josh interviews Dr. Geoffrey Shapiro. Dr. Shapiro graduated with a doctorate of medicine from Cornell University in New York. Go Big Red. He completed his oncology training at Dana Farber and joined the department faculty in 1994. Dr. Shapiro's particular area of expertise is early phase clinical trials, and he currently heads the Centre for Cancer Therapeutic Innovation, with a special focus on cell cycle and DNA repair inhibitors.
Hi, Jeff. Thanks so much for being on the show.
Sure. Thanks for having me.
So, Jeff, what I'm trying to do with this sort of series of episodes is kind of talk about your journey, how you ended up being at Dana Farber, your work in the lab and those sorts of things. Can you take me back to the start of your career? What led to you getting involved in oncology and ultimately ending up with becoming a clinician scientist?
Oh, well, going back quite a long time. All right. So when I was a resident in the late 1980s down the street at Beth Israel. And, at that time, on my oncology rotations, I saw it as a field of tremendous need. It also seemed like a field where there was exciting research and the ability to really provide very close and intimate care to patients. And so those two things seemed to come together for me. And so I decided to pursue oncology fellowship. And then I went into medical oncology fellowship back in 1991. I did that here at the Farber for a few years. And then I suppose you could say that I'm one of those Farber boys that never left. And I had good experience during my medical oncology fellowship and had a very well-rounded experience treating patients with many different types of cancer. I then did some work in the laboratory for several years, and I worked on cell cycle related genes in lung cancer. And that actually spurred my interest in cell cycle therapeutics. And I began to develop drugs that were related to the cell cycle. For quite a while, I was in thoracic oncology from my clinical work, but as new drugs were being developed that were related to my research, it seemed more relevant to be in the phase 1 program where new drugs are first developed. So I guess around 2000, maybe about 23 years ago now, I moved over to the phase 1 program at the Farber. And then I've really been there ever since. So I led the phase 1 program here between 2007 and 2020. And have developed many cell cycle and DNA repair inhibitor drugs within our program, and enjoy working on the early phase studies and providing patients with new opportunities once standard treatments have failed.
And going back just a little bit, that was probably 30 plus years in the making in 15 seconds or less. With your PhD, so I know you've done a PhD and that was in, I think, cell cycle as well. What sparked your interest in that specific area? Oncology going back 30 years was a very different game.
Yeah. No, for sure. So actually, I did my PhD more in molecular biology and actually worked on influenza virus. So when I first was in medical residency, I thought that I might specialize in infectious disease. But again, it really sparked my interest was the complexity of oncologic diseases when I was doing rotations with medical oncology patients, and the ability to be their primary doctor also attracted me. And that was the two primary reasons that I pursued oncology fellowship. At that time, it was a very exciting period at the Dana Farber where there was many new aspects of the cell cycle that were being discovered. the role of the retinoblastoma protein, the ability to phosphorylate the retinoblastoma protein to move the cell cycle forward, the roles of cyclin-dependent kinases and CDK inhibitors in cell cycle progression as universally dysregulated in cancer. So I saw that as a very, very important and critical pathway and wanted to begin to try to do translational work in that space. And that is really how my developmental therapeutics career got started.
Amazing. You have over 671 publications, give or take, maybe more, after doing a little bit of a deep dive.
Well, that might include abstracts as well. So for full manuscripts, it's probably between 200 and 300. So maybe about half of that.
Okay, sure. Look, either way, that's like one a week. I'd love to talk to you about, where you see the role of the phase 1 team going, and also how you'll manage the complexities of sponsor-driven trials with funding and both patient expectations and toxicities. Could we go into that sort of a ethical question?
So phase 1 work has really evolved over the years. So previously, phase 1 studies were really very small studies, usually first-in-human compounds. They were given in unselected ways to patients. They were new drugs coming off the shelf. We didn't really have biomarkers for selection. And patients who had failed standard chemotherapies that wanted to try something new would go into these small phase 1 studies. Phase 1 studies, develop drugs starting at very low doses. Small groups of patients will get increasing doses with each group of patient until we figured out the highest dose that we can give safely and what might be a dose that's used then in further line efficacy studies. So much about the landscape has changed, really, I would say, over the last 10 to 15 years. So the first issue is that we try to be a little bit smarter about what drugs that we give to patients. So with the advent of tumor profiling, we now have a better understanding of drivers of patients' cancers. Most patients now will come in with some targeted next-gen sequencing and profiling of their tumor, whether it be profiling of the tumor cells itself, or now in this day and age, even a blood test where there's a liquid biopsy that just gives us some idea of what the driver of the cancer might be. Of course, lung cancer is the big field where this, one large field where this all started, where really based on the mutations in the cancer, one can identify drivers and then couple them with the correct drugs. But we do try to use the profiling to be a bit more intelligent about the drugs we offer. The other issue that's occurred is that these studies have gotten quite a bit larger and quite a bit more complex. So first of all, we often don't stop simply when the maximum tolerated dose is achieved. Often there will be additional work in subsets of patients before it goes on to larger studies. So a phase 1 study becomes a bit larger and more complex. And when there are subsets of patients enrolled, often those are by disease type. And so we have to have a very facile interaction with disease centers between the phase 1 program and the people who are disease specific, so that studies can move forward efficiently and move quickly from the phase 1 dose escalation into potentially specific disease types. So a lot more interaction, and phase 1 no longer really exists in a vacuum. The other thing that most phase 1 programs, always in the past, we always studied pharmacokinetics of the drugs and the way the drugs were metabolized with many research tests and following the level of drugs in the bloodstream over time. But now these studies also have really more proof of principle and proof of concept studies that are associated with them. So we often obtain pre and on treatment biopsies in patients, and we look to see whether we've engaged target. Is the drug hitting the target? It is behaving on the cancer cell the way we expect it to behave? And does all of that biologic effectiveness of a specific drug ultimately correlate with the clinical outcome? So the studies have become quite a bit larger, more complex, difficult to do. Patients, for patients to participate in these studies, the research requirements have escalated quite a bit. Patients give us quite a bit of time and courageous volunteerism in order to participate in these studies. And because these studies are complex, they're often shared among multiple cancer centers. So a single cancer center never really does just a phase 1 study anymore. So many phase 1 studies are shared over multiple cancer centers, and there has to be very, very good interaction between the investigators and the centers in order for the study to move forward efficiently. So it becomes a very, very complex entity and a very, very complex enterprise to maintain phase 1 programs. There are also studies that are phase 1 studies that are directed towards specific mutations that patients' tumors might harbor. But often those mutations might be rare alterations. And so that requires quite a large number of sites to participate in order to enroll patients with the correct alterations. So phase 1 becomes a very large enterprise, a collaborative effort, and very, very complex. So the study teams that are leading these studies have to be somewhat large. We have to have all sorts of regulatory help. We have to have research nursing help, clinical pharmacy help. Phase 1 programs are really quite extensive programs in this day and age in order for these things to move forward efficiently and move forward with generating the robust data that we need to know whether drugs should continue to move forward.
One of the concepts that was raised recently at a conference here in the US is the idea of the maximum tolerated dose.
Yes.
In the phase 1 world. So, dose limiting toxicity, finding the maximum dose versus efficacy. Do you think in the field moving forward, this might actually start to shift rather than finding a better balance from our drugs?
Yes. So this is the Project Optimus that the FDA has espoused, and that the dose defined by the highest safe dose we can give or maximum tolerated dose might not always be the optimal dose moving forward. And so when should we optimize that dose? So one can do a phase 1 dose escalation and get the maximum tolerated dose. But if we've done some biopsies along the way, we might recognize that a lower dose might look like it has some biological effectiveness. And so should the lower dose be compared to the higher dose and really understand whether we need the higher dose to really move forward in larger studies? And so Project Optimus would have us stop at the end of a maximum tolerated dose and potentially randomize that maximum tolerated dose to a lower dose, see if the lower dose is equally biologically and clinically effective. And maybe that's the dose that should move forward for further development and ultimately what goes into large groups of patients. So this has actually gotten to be a very complex question because one could take time to do that randomization early on and see if we've optimized, try to optimize the dose early. But what happens if the lower dose turns out not to be as good? Have we expended using that dose in a group of patients who maybe should have gotten the higher dose all along? And should we potentially maybe take our highest dose, get it into the pivotal trial so the drug is FDA approved and available for a large number of patients, and then go back later potentially and do some optimization and see if a lower dose could be equally as effective? And so there's quite a bit of controversy and debate not about optimization, but actually maybe more the timing of optimization. And should it be done earlier in the drug development process, or is that going to unnecessarily delay getting the drug to large numbers of patients? And should we potentially go back and do that optimization later? So lots of discussion about this. The FDA has called many meetings and workshops, and there's a lot of discussion about what's going on. But there's no question that simply obtaining the MTD, the maximum tolerated dose, and assuming that we're all finished and that there should be no further dose optimization, that concept's going away. And I think everybody realizes that doses probably need to be optimized based on ultimately what is a totality of data. Not only the highest safe dose, but what is a biologically effective dose? What is tolerated best over time? When we define an MTD, that's only often over the first month of treatment. So what is the real tolerability over long periods of time, especially when patients stay on targeted or immunologic agents? So complex questions that do make drug development exciting and interesting, and definitely keep us thinking for constantly about the best way to do this for patients.
So can you tell me a bit of you've done a lot of phase 1 over the last 23 years. What are some of the pivotal trials that you've been involved in that have led to kind of practice changing or paradigm shifting new drugs?
I wish I had more of them, but I do have a few. So I did quite a bit of phase 1 work on the cyclin-dependent kinase inhibitors. So we did really 3 pivotal phase 1 studies on abemaciclib, ribociclib. We didn't do the actual initial phase 1 of palbociclib, but we did the first study of palbociclib that actually demonstrated it hit target. So some of the story with palbociclib or Ibrance for breast cancer really was that the phase 1 study it actually went on by some of my colleagues in the phase 1 field, had completed the study, and there were 70 to 80 patients enrolled. There were doses that were established actually on 2 schedules, but there wasn't a whole lot of activity. There wasn't a lot of shrinkage of patients' tumors, and there wasn't a lot of what we call partial responses. So Pfizer was really thinking about putting the drug on the shelf, and we actually convinced Pfizer to let us study palbociclib, not in breast cancer, but a different tumor called mantle cell lymphoma. And mantle cell lymphoma is a cyclin D1, CDK4-driven tumor. The translocation, which defines the disease, is a cyclin D1 translocation. And so it's driven by CDK4 activity. And we used palbociclib in mantle cell lymphoma, and we said to Pfizer we would get biopsies and do other pharmacodynamic work to at least demonstrate that the drug hit target. And so we did actually the first study that demonstrated that palbociclib was actually hitting its target in tumor cells. So we did that by biopsying tumors before and during treatment, and we demonstrated that there was a reduction in the phosphorylation of the retinoblastoma protein. We worked out phosphospecific antibodies that we could use immunohistochemically, and we demonstrated that the drug really shut off CDK4 activity. And then we used a special type of PET scan called fluorothymidine PET scan, which demonstrated that a consequence of that was to reduce the fluorothymidine PET uptake in the patient's tumors, which was consistent with G1 arrest. And some of this did actually correlate with good outcome for some of the mantle cell patients. And so Pfizer was convinced that the drug was hitting target, and then was a little bit more liberal about letting the drug go forward in larger studies. And the drug began to be studied more extensively in breast cancer. Some of that was happening simultaneously, but to begin to be studied in larger breast cancer studies and combined with hormonal treatments. And then also it was studied in a disease called liposarcoma, where actually the drug now has, and by the NCCN guidelines, palbociclib can be used in liposarcoma. So we did some of the first study that really demonstrated that palbociclib hit target, and that shutting down the cell cycle in these mantle cell lymphoma cells could actually correlate with a good clinical outcome. We then went on and we did do the phase 1 studies. I was involved in the phase 1 studies of both ribociclib and abemaciclib, drugs that ultimately then were combined with hormonal therapy and have made a major impact in breast cancer. For abemaciclib, we used our retinoblastoma phosphospecific antibody assays again, and we actually demonstrated that if we only gave the drug once a day, we didn't maintain target engagement for the full 24 hours, despite some of the pharmacokinetics that we were achieving with the drug. And so that is actually what converted abemaciclib to a twice-daily schedule, where we showed with the twice-daily schedule, we could suppress the RB phosphorylation for the full 24 hours and get better target engagement that was lasting. And that actually contributed to the twice-daily scheduling that we currently use for abemaciclib. So I would say for those 3 drugs, for palbociclib, abemaciclib, and ribociclib, the drugs that have ultimately transformed breast cancer care, I think we were involved with very pivotal early phase studies, which helped those drugs move forward. So I suppose I'm very, very proud of that work.
Amazing.
Yeah.
You might be able to answer this from a phase 1 perspective. So the data readouts of ribociclib, palbociclib, and abemaciclib not all come to fruition, and they've all got somewhat varied results, with palbociclib almost being the poor cousin, depending on who
as the data have evolved, yes, over time.
Was there any signals in the early data or in the early phase 1 stuff that you could see that might have indicated different binding to the CDKs or sort of different efficacy?
Yeah, so there was always a question about whether abemaciclib was a bit different. So abemaciclib clearly behaved like a CDK4/6 inhibitor in many preclinical models, but biochemically, the drug did seem to be a tad more promiscuous in vitro assays, potentially also hitting CDK2 and also potentially hitting CDK9. So that one potentially did look that it could have been a bit different biochemically from the beginning. At the clinical level, abemaciclib also had a different toxicity profile. So whereas palbociclib and ribociclib were more hematologic toxicity as the primary toxicities, ribociclib carried a bit more baggage as well, but mostly hematologic toxicity. Abemaciclib seemed to have a bit less hematologic toxicity and more gastrointestinal toxicity. And it's not completely clear why that is and whether it is because it might hit some of the other CDK proteins. But there were some hints early on that abemaciclib might be a little bit different. And some of the emerging data now is suggesting that abemaciclib might be useful after, say, palbociclib. So some of the data that has been developed by our colleagues at Mass General has demonstrated that we might be able to use abemaciclib even after palbociclib. Same, though, is beginning to be true with some of the data coming through on ribociclib as well, which is a little bit a little more challenging to understand, except if one can argue that there might be slightly different potencies and a slightly different ability to inhibit CDK4 a bit better. So we don't know the full answer to the question, but it's true in the overall survival data, the drugs are not looking absolutely identical. And this is affecting practices right now as the data continue to emerge.
It was a curly question, I must admit. I don't think there's a golden answer to that. I have a couple more questions. One being, from an American standpoint, equitable access to trials with balancing time toxicities and financial toxicities. What do you see the role of phase 1 in playing this? Because in Australia, there's quite a big conversation now. It's like we define time toxicity as number of encounters with the healthcare system on any given day. And phase 1 definitely has significant requirements from a safety standpoint. How do you see this evolving?
Right. So I think there needs to be very good partnership between insurers, the federal government for NCI-sponsored studies and drug companies. But I think we have to have coverage for patients to participate in clinical trials. So typically the way trials work is research tests, studies that are only done for research need to be paid for by the sponsor, which would be the pharmaceutical company or in some form of an NCI grant if this is an NCI-sponsored study. And then there will be other components of the patient's care that could be considered standard of care. And we have to have insurers cooperate in order to give people access to trials. So there has to be partnerships from many stakeholders and many components. But ultimately, there has to be equity, there has to be access, and everyone needs to be able to access these studies. One of the things that's hard, though, is that in the early phase studies, there's so many research requirements that often these sort of get done at the larger cancer centers. And the question is there anything that we can do to make things easier for patients and allow a partnership even with community oncologists to let more things be done locally, potentially so patients that are not in close proximity to a major cancer center can still participate. So some of this was actually evolving during COVID, where we were able to ship drugs. Patients were able to do laboratory tests or evaluations with local physicians, and that still was acceptable data then to use. All of that has kind of gone away as the pandemic has abated. And I wonder whether some of those things that were evolving are things that should be thought about again in order to make trials more accessible and easier for patients to participate.
Yes, the role of teletrials and remote access is a very important thing. For young researchers who are interested in the phase 1 world, what role do you see them playing in the future? Well,
I think we always need new blood. And we always need new physicians coming in that are interested in developing these studies and working in them. It's interesting, though. Medical oncology still really does work by disease types, and people, especially at academic medical centers, do tend to specialize in disease types. But in each disease type, one can enroll patients that are on phase 1 studies. So the way we work it here, for example, is our phase 1 program has early career investigators who are liaisons to the phase 1 program, even though they work particularly in one disease center. And so there'll be one person in thoracic, one person in ovarian, one person in breast. Breast cancer, they become expert in those fields, but they're also then the person that has the pulse on the new trials, what's coming through phase 1, and are able to offer those trials to their patients. And so I think that's more of the interactions that the phase 1 program has to have with the disease centers. And that's a good way for an early career investigator to think about doing it. Maybe having some disease specialization, but also understanding the phase 1 offerings and be able to offer them those trials to those specific populations.
And one final question.
Sure.
what words of wisdom would you give your younger self? Can be personal, professional, life, research, doctoring, in general. What would that be?
Well, that's a very good question. So I would say that, I guess I've always been a 50-50 person, but so I've tended to try to do laboratory work that then feeds the clinical trials. But I also so I do feel that that's been a very satisfying component of my career, to actually have the laboratory do basic and translational work and then try to translate that to a specific study. My work did become a bit unfocused, though, when I sort of more broadly became involved in phase 1. So I was doing many phase 1 trials, some of them emanating from my work and some of them not so much. Was more just to be involved in a multi-center phase 1 trial and have that offering for patients as part of my clinical effort. I think ultimately that component of the work might have been distracting. It's not that it wasn't important. It's not that it wasn't good to have those options open to patients. But I think that my feeling would be that very, very clear and very, very laser focus is a better way to succeed and get more impact. And so I might have limited the number of trials that I did and maybe focus more on very specific modalities of trials. For example, the CDK inhibitors, or another group of drugs that I work with, the DNA repair inhibitors, and maybe not have such a global portfolio of trials that maybe detracted from my focus a bit. So I think if I gave some advice to my younger self, it would be to maintain the laser focus on the field of interest and that you believe in and the pathways that you feel need to be disrupted, and to make sure that those grounds are covered before taking on too many other things. I think the other thing that I wish that I had been a little bit faster to recognize would be the impact of immuno-oncology. And so if I had any maybe advice to give to myself back then, it would have been to become more schooled on the immune microenvironment and on the immunology of cancers a bit more. And I might have been more prepared to understand some of the interactions of our targeted agents with the immune microenvironment as this field moved forward. So I was, late to the field of immuno-oncology. I'm still learning a lot of immunology now, and potentially I wished I had mastered that a little bit sooner.
Fair enough. And I know that I said that was the final question, but I have one more final. This is the final. What does it take to be a top doctor in the United States?
Oh my. I'm not sure you're talking to one, but no, I think that, you have to have, I think you have to have very good training. You have to have good mentors along the way. I think that as far as oncology is concerned, I think getting trained in both clinical research, patient care, and basic translational research, exposure to all of that is very, very important in order to be able to master the field and stay on the pulse of the field going forward. So I think a well-rounded training with very, very good mentorship is a good set of ingredients that will ultimately help people develop into becoming top physicians. Of course, one can never forget the patient as the focus of one's work, always maintaining one's compassion and advocacy for patients. And I think that's also another critical component of maintaining oneself at the top of the shelf for being a top doctor.
Thanks so much for being on the show.
Sure.
Thank you for listening to this episode of Oncology for the Inquisitive Mind. You'll find links to the rest of our episodes on our website, inquisitiveonc.com. There you will also find a collection of weekly blog posts, useful resources, as well as links to our Twitter and LinkedIn pages. This is Oncology for the Inquisitive Mind, a podcast by ADC Productions.